We will now look at skeletal muscle metabolism. The energy necessary for muscle contraction is provided by ATP. Because the muscle stores a limited amount of ATP, mechanisms must be in place to synthesize new ATP quickly to enable continued muscle activity. Your goals for learning are to understand the cellular processes for synthesis of ATP, to compare and contrast aerobic and anaerobic processes in the muscle cell, to examine the differences in ATP synthesis among different types of muscle cells. Here's what you need to know. The events of a single cross-bridge cycle, definitions of hydrolysis and dehydration synthesis, the role of enzymes in cellular processes. To review the events of a single cross-bridge cycle, click the Link button. If you use a link button, you can return to the page you started from by clicking the Return button. To see definitions of terms, click the bold red words. In muscle cells, the energy molecule, adenosine triphosphate, ATP, plays an important role in 1. Energizing the power stroke of the myosin cross-bridge. 2. Disconnecting the myosin head from the binding site on actin at the conclusion of a power stroke. And 3. Energizing the calcium ion pump, which actively transports calcium ions back into the sarcoplasmic reticulum. ATP is composed of the adenine nucleotide with 2 additional phosphate groups attached by high-energy phosphate bonds. The potential or stored energy in ATP is released when the terminal high-energy bond is broken by a hydrolytic enzyme. The end products of the hydrolysis of ATP are ADP, adenosine diphosphate, inorganic phosphate, and energy. Click the button to see hydrolysis. The end products of ATP hydrolysis are not discarded as waste, but can be recombined to form a new ATP molecule. Rebuilding ADP into ATP requires a synthetic enzyme to carry out dehydration synthesis and a new source of energy to rebuild the high-energy bond. Click the button to see dehydration synthesis. When ATP is formed by dehydration synthesis, it's like money in the bank. In fact, ATP is often called energy currency. When muscle cells hydrolyze ATP, energy is released, and, like currency, can be spent for moving myofilaments and transporting ions. Just like our piggy banks, ATP currency is limited. Muscle cells have only enough ATP to last for a few seconds during an active contraction period. Click the muscle cell to begin contraction and spend the ATP energy currency. Now that the ATP supply has diminished, let's look at how ATP is regenerated. When ATP supplies are low, muscle cells use three processes to synthesize additional ATP. 1. Hydrolysis of creatine phosphate. 2. Glycolysis. And 3. The Krebs cycle, also called the citric acid cycle, and oxidative phosphorylation. Click the creatine phosphate box to see its role in ATP synthesis. Now that the ATP supply creatine phosphate, an energy storage molecule, is the immediate source of energy for rebuilding ATP. Substrate phosphorylation is a hydrolytic process that transfers energy and a phosphate group from creatine phosphate to ADP, forming ATP. The amount of creatine phosphate is limited and is rapidly depleted in active muscle cells. While the muscle cells are using up creatine phosphate, they must turn to another source of energy, glucose. Click the blood vessel to learn more about glucose. Glucose is a major source of energy for synthesizing ATP. Glucose is available to muscles from two different sources. 1. Glucose enters the muscle cell directly from the blood. 2. Glucose is produced by hydrolysis of glycogen stored in the muscle cell. Glucose is broken down in a process called glycolysis. The end products of glycolysis include 2 ATP molecules and pyruvic acid. Pyruvic acid can follow one of two metabolic pathways. Click the pyruvic acid triangle to see the first pathway. Glucose is broken down in a process called, in the absence of oxygen, pyruvic acid is converted into lactic acid. Lactic acid is the end product of the anaerobic pathway. During continued anaerobic conditions, excess lactic acid builds up rapidly, bringing about muscle fatigue. To avoid muscle fatigue, oxygen is needed. Oxygen is available to muscle cells from two different sources. 1. Oxygen enters the muscle cell directly from the blood. 2. Oxygen is stored in myoglobin, an oxygen-binding protein that is abundant in certain types of muscle cells. To see the aerobic pathway that takes place when oxygen is present, click the Krebs cycle and oxidative phosphorylation box. Oxygen is available to muscle cells from two. When oxygen is present, the aerobic pathway will proceed, that is, the Krebs cycle and oxidative phosphorylation. Pyruvic acid is converted into acetyl-CoA, which enters the Krebs cycle in a mitochondrion. Energy is transferred to ATP in the process of oxidative phosphorylation. The end products of the aerobic pathway are carbon dioxide, water, and 36 molecules of ATP per molecule of glucose. The total energy harvest from glycolysis, the Krebs cycle, and oxidative phosphorylation is 38 ATP molecules for each glucose molecule metabolized. Although not shown here, the metabolism of lipids and proteins can also be used to synthesize ATP. The immediate source of energy for synthesizing ATP comes from creatine phosphate. The second source of energy for ATP synthesis comes from glycogen and glucose during the process called glycolysis. The end products of glycolysis are 2 ATP molecules and pyruvic acid. If oxygen is not available, pyruvic acid is converted to lactic acid in the anaerobic pathway. In the presence of oxygen, the aerobic pathway takes place. Pyruvic acid is converted into acetyl-CoA, which enters the Krebs cycle. In oxidative phosphorylation, energy is transferred to ATP. The aerobic pathway yields the greatest amount of ATP, 36 molecules of ATP per molecule of glucose. Byproducts of the aerobic process include water and carbon dioxide. Recall that ATP is used by the muscle cell for the power stroke of the cross bridge, for disconnecting the myosin head from the binding site on actin, and for transporting calcium ions back into the sarcoplasmic reticulum. The mechanisms that produce ATP can be compared to a cellular factory. To see animations of these processes, click the ATP Factory Tour button. To skip the tour, click the Factory Bypass button. Here's a summary of what we've covered. ATP must be synthesized in muscle cells to replace the ATP used for muscle contraction. ATP is synthesized by hydrolysis of creatine phosphate, glycolysis, and the Krebs cycle and oxidative phosphorylation. White muscle fibers mainly use glycolysis for synthesizing ATP. These fibers are quick and powerful, but fatigue rapidly. Red muscle fibers mainly use the Krebs cycle and oxidative phosphorylation for synthesizing ATP. These fibers are fatigue resistant and have high endurance. To test your knowledge, click the quiz button to go to the self-quiz.